There we go. Hey, everybody. All right, we missed you on Thursday, so let's get into it today. I had to testify as an expert witness in a court case, so we'll talk about that just in broad terms today. On the basis of being a researcher in a particular area, so.
It does.
It was stressful. Oh my gosh.
Oh, I wasn't gonna testify in my daughter's best friend's custody.
Okay.
Okay, I'm like,
I got deposition and had to testify in court when the girl got hit. The girl on a bicycle got hit by a car several years ago on
Oh.
campus.
Yeah, okay.
Yeah, because Peter Dirks and I saw it happen.
Okay.
And that went on for years. It was like 3 years when we finally testified.
Yeah, this court case is the same way.
it's
They
just
give you
so
your, what,
intense.
$10 or $25?
Yeah. But it's nerve wracking.
It is. Peter just looked at him, Peter, he just looked at him and said, she was gonna get hit, whether it was this car or the next car.
Just
Gonna
like-
ride a bicycle in the ground.
Blasting through the crosswalk or whatever.
Yeah.
Yeah.
Well, no, it wasn't even a crosswalk. It was just on right at the railroad tracks they ran. She
Oh,
was-
yeah. Okay.
I guess she thought the double line was a crosswalk, but it wasn't. Just
Oh,
drove right into the path of traffic.
wow. Okay. That's
Stupidity. I don't know what you expected.
That is intense. All right, well, let's get this screen sharing set up. I think it's about time to get started here. So let's do our thing.
It's big over there. There were several students. I saw several of our students. There's several forestry companies too that I talked to.
Good. Was it good turnout this year?
Yeah.
Awesome.
We have a lot of companies up there.
I'm getting a lot of questions about students, for summer employment, stuff like that. So this is
There's a lot of
perfect
summer
timing.
internships still left over there.
Good.
Timber products, he's an alum. He said he was here early this morning and there wasn't a lot of activity in the building. Like, yeah, this generation's a little bit different than when you were here.
There we go. All right. Okay, so let's kind of recap last week a little bit, and we'll talk again just briefly about the court case and stuff like that. I'm not supposed to talk in any sort of specific terms, but we'll just regard it from more a professional perspective because each of you is studying to become an expert in your particular area of expertise, and so it may come to pass that it is important that you are really pretty sure of what you are saying as an expert. And hopefully your expertise is helping prevent court cases. Man, it's a shark tank. Okay, so last week we were talking through kind of different stages in the life cycle of a research project, different stages in the generalized cycle. It's very much a cycle of the scientific method and performing research. And there was particular emphasis on noting, hey, this also really applies if your research is not lab-based, if you're doing documents research and your analyses are synthetic, you are pulling together a lit review and finding what is important out of that, interpreting what's important out of that, identifying what is missing in terms of what's known, and therefore really important for immediate next steps in your particular area of expertise as well. So we don't want to make the sin of omission for excluding anything that doesn't look like a lab experiment. It all counts. This is all science, just different faces of it. Okay, so that was Tuesday's class. On Thursday, again, I was out giving a deposition, so it's not in a courtroom at the moment. And the basics of the details of the case don't particularly matter. What was important for me to experience that I can warn the rest of us is that when you start amassing some advanced credentials, professional level degrees like master's, PhD, whatever, people really do start to look to you as an authority, as a source of truth and knowledge within your domain of expertise. And in some cases, in spots where it might get out towards the edge of your expertise or off just past that boundary. And it's important on an ethics basis. We have an ethics module in this class. We'll get to that later. It's important to be very clear with yourself and then also with the lawyers involved, because there are real people's lives wrapped up in this stuff. Hey, I'm really good at this part of it. I'm providing informed opinions on this part that's pretty adjacent, and I'm not really comfortable stating definitively this dynamic that's related, but it's not my area of expertise. A, because the lawyers will smell blood in the water and tear you apart, and B, because a lot of the momentum of a case is based on how confident, how sure are you of these things that you're saying and speaking into the case. So it's the same kind of dynamic as, if you're working for NRCS or Farm Bureau or whatever, and, a farmer's coming to you asking, hey, what do I do about this particular situation? You don't want to overgeneralize your expertise and try to apply to something where you're genuinely not as confident or sure. But where you are, work hard to translate your expertise into terms that, a lawyer or a farmer or whomever else you're working with, in a city water district, if you're doing water research, translate what you understand, what you're seeing out of the data in a functional way to them. So part of this class is science communication and being very much a translator or interpreter, because what you're saying at a full professional technical excellence level is totally Greek to pretty much everybody else. And that can do some harm as well as good if you're not doing the translation, okay? So, this is something to keep in mind for the assignment, to go check out a couple of seminars across campus, dissertation or thesis seminars, or invited guest speakers or whatever. Are they translating the jargon to an intelligent audience, but not one that is necessarily versed in all of their jargon and vocabulary and specific nuances and dynamics? When you're given your seminar on campus, you can assume that pretty much everybody in the room is, not a complete idiot. You might have somebody asking you irritating questions in the question and answer session. You might consider them an idiot, but that's separate. And there are techniques to deal with that as well. But is the person recognizing the fact that the point of this is not to show off how smart you are, it is to transfer meaningful and valid and accurate knowledge and interpret what's important out of it? Or is the person just kind of sounding off and kind of failing to connect, if that makes sense? So again, that's kind of the last third of our semester, working on some of those techniques. So we'll get there. Any announcements for the good of the cause? Any seminars on campus? Interesting things going on. There's the job fair, of course, or the career fair. Other events?
Flyer I'm going to bring with it with the ad on February 23rd, where they're bringing that lady who does the water.
Yeah.
Flyer.
I think I wrote that down. Art of science, something.
She's a really top. Expert in natural research.
Okay, yeah, send it on over. I'll post it on the course website and we'll get to it. So back to, there we go. All right, so what else? Outlines, literature review, annotated bibliography, bibliographic citation software management, How's it going? Let's check in.
I met with the librarian who recommended I get EndNote, so I actually got approval yesterday and downloaded it.
EndNote. All right.
Don't know how to use it yet, but we'll see. But Mary Taylor highly recommended. She said you need to get this. If any of you need the librarian, I'd highly recommend scheduling a Zoom meeting with her. She spent an hour and a half with me, just showing me different searches and best places to find. She'll send you articles as she finds them, even journal articles that would relate to your research. So.
All right. So if you're not able to hear this on Zoom because the mic's aimed the other way here, our research librarians are glorious. Spend an hour and a half with you, and that is very expensive otherwise. You know, this expert consulting can run a couple of $100 an hour. So.
And she's always there to help. So if you run into a problem, she'll schedule time with you again. So it's just open-ended.
Excellent.
And I loaded my outline for the first time last night. So I'm trying to get a committee meeting together, but I have to send out a meeting to the call.
Okay, cool. Very good. Let's kind of, I guess, go around the classroom. So how are things going with the literature review?
I'm reviewing limits. That's where it's going. You know, I took statistics, reading some of the, journal articles when they start with the little signs and the little, I'm just like, what? So
Okay.
it just takes me a while.
That's valid. All right. So just repeating for folks on Zoom. Literature review is going okay. It sounds like a pretty big stack of journal articles, and sometimes the stats kind of make your eyes glaze over a little bit and so forth. How many here in the class and on Zoom tell us a little bit about your stats backgrounds so far? Has everybody had a chance to take like the mandatory intro stats class yet, or is that still kind of on the docket for folks?
You mean like the graduate level?
Yeah.
No, I'm taking it in the fall.
Taking it in the fall.
I'm in the second semester.
Second semester. Awesome. Same. All right. Fall semester.
I took a form 502 and my instructor's name was really good. Well, I mean, he would get real frustrated because he's really smart and people wouldn't do that. But he explained things really well. So
Good. All right. I had one of the stats classes, one of the many I had to take as a PhD student, was taught by a fellow who absolutely knew his stuff. And he had a pretty thick accent, which was a practical difficulty. And he was energetic and very happy to take time to explain things like outside of class, which we ended up having to do a lot. But yeah, it can go well, or it can just, everything is a little bit harder when it's difficult to hear folks.
That's the same experience. I mean, he's from Africa.
Okay.
But he would start saying, and we'd be like, "Wait, back up. What was that word?" He's like, "Oh, you're all wrong."
All right.
But he was good.
And I made it through 506 last fall.
Okay.
So I have to do 507.
All right.
Dr. Pence recommended I wait to do it for my own research.
Do we have to do both? Or is it's just the one that's required, right?
I
Let's-
have to do both. in the future.
yeah, for master's students, I think it's just one, right?
Yeah.
I wish it was
Yeah.
just one.
Yeah. So
But that's
you're
what
out
it said
for-
on the website anyway for forestry, so I hope that's still true.
But my question is like, which department? Because I think there's like 5 different stats classes in different departments.
Yes.
What stats, like there's biostats?
I
there's
think
social
there's
stats.
just the one that you're expected to do for grad school, and I think it's just in like biology or something like
Biostats
that.
that,
But it's like 506.
Yeah, you're allowed to, I believe you're allowed to select, or at least you used to be able to pick which one so that it was anything closer to your particular content area. So for example, biostatistics is going to have a lot of habitat and zoology and biology statistical measures. And, everybody's going to talk about, mean, median, mode, standard deviation, range, variance, like the basics that you would get in a first grad level stats class. But the examples tend to be customized for one or the other. So there's a grad stats class, Quan. There's an ed psych graduate stats class, which tends to deal more with things like surveys, pre-tests, post-tests, and human measures. And I'm sure there are plenty of others as well. And then everything past the 506, 507, 1-2 sequence rapidly becomes more domain specific. So there's, geospatial statistics, there's a sequence of those. There are like proteomics and genomics is a subset of biostatistics for genes and proteins, and so forth and so on. So even as a master's student, if you don't hate 506, 507, it's never a bad idea to take a more specific class that's going to deal with the stats that you are planning on using. Generally, in my experience, the required 506 level class is it's a good starter, but because it's so sort of foundational, I mean, standard deviation is like pretty early in the journey of statistics. And most of us are going to be doing something beyond standard deviation. Not all of us, right? So this doesn't have to apply to everybody. It's not a one size fits all sort of thing. But if you have the fortitude of soul to take an additional stats class at the graduate level for your particular content or topic area, that can make both the mechanics of grinding through stats a little bit more understandable, and it helps you race ahead, in my experience, in terms of understanding why and how you're doing what you're doing. Yeah, stats it's a whole sort of worldview and mindset. You have to sort of train yourself to think like a statistician. Most of us are not born that way, and that's okay. That's what these classes are for. And if you're just not a math person at all and you're just hating life through the 506 class, just get through that and, maybe work instead a little bit more closely with your advisor who can, and your research group, your fellow grad students who are probably doing things in the same domain area that you are. That can get you through on the stats that you need for your project pretty well as well. So you have a couple of different options to customize and pick from among what you need. Okay.
I just didn't think I could do a back-to-back semester. I was so over it after that. I'm like, I can't do this.
I hear that. And in chat, we've got it's advanced biostats now. All right. Elaine, hang in there. You're still typing in coherent sentences, so I think it's going okay, I hope. And Maggie took it in 2016, I think that says. It's a little bit blurry on the screen.
Yeah, it's 2016.
Awesome. Okay.
I just emailed you all that forestry, that water.
Okay,
that
cool.
webinar thing.
All right, so I'll post on our course website a link to that. Thank you for doing that. All right, so our sense of, let's get this other PowerPoint closed off here. There we go. So our sense of what's going on with our class discussion here is, shoot, let's see if we can close that chat box. Oh, no, we can't. All right, hang on. Zoom troubles, just a moment. Moving on. Let's get this started again. Sorry, the mouse was going behind that chat box. And I can't work like that. Okay, let's see. So from a statistics basis, again, not all research projects are stats based. We do also have meaningful and useful and valuable qualitative studies as well. But our current slide here, let's get that shared again. Zoom. There we go. All right. So I've taken a moment to kind of break out what are the typical stats that you would start to use. These are very introductory, very general. What kind of stats tools would you use? Some of those stats tools are going to be tests where you accept or reject or more technically, reject or fail to reject your hypotheses built out of your research question. So descriptive research it's generally univariate stuff. So if we're talking about a population of people as our unit of analysis, human individuals, that's going to be, at the first blush, univariate statistics. What's the distribution or the histogram, which is a chart of the count of each value of people's height or body mass, or what's the frequency table, so the accounting of different eye colors, or hair color, or hair texture, whatever. Height and weight are expressed in terms of just themselves. What's the average of that? Univariate summary statistic. What's the range of it, right? Maximum value minus the minimum value. The variables distributions. So in your stats class, if you are taking it in the fall, or you've already taken it as early as 2016, distributions summarize, you get a graph of, typically is a line graph or a column chart of what's the count of the values and what's the shape of that count, if that makes sense. So we're all probably familiar, even if you haven't taken a stats class yet, of what's called a normal distribution. So it's a symmetric hill shape, and the peak of the hill is the most common value in that set, and it tends to be in the middle of the value of the distribution when you are measuring things in nature. So the normal or Gaussian distribution is very, very common in nature, and you can describe a few kind of stats that are derived from the characteristics of that particular distribution. Like if it's a really symmetric peak, but it's a really fat peak, like there's less clustering, or it's less common to get exactly that middle average value. There's a statistic that talks about what is the wideness or the fatness of the Gaussian distribution. It's called kurtosis. And so you can have a very skinny, sort of like a needle-like peak, or you can have a very wide one that has more values out to the extreme ends, low or high, around that middle average value. Univariate statistics are pretty straightforward, and most of us have been familiar with the concept of average for a really long time. Statisticians have complicated univariate stats just a little bit by adding other ways to describe things like central tendency. So average, arithmetic average, as we understand it, is one way to describe central tendency. It's kind of vulnerable to, for example, if you have one really crazy outlier. So let's say with height of humans in the population you're studying, everybody in the United States, average height for a male adult is about 5 feet 10 inches. And we've got a couple of people that are, below 4 feet tall, maybe achondroplasic or dwarfism or whatever. We've got a couple of people who are way high above that, Marfan syndrome, like Abraham Lincoln had, giantism, stuff like that, where growth plates don't close, or pituitary tumors or whatever. But generally speaking, if you had to pick somebody at random, they're most likely to be, if it's an adult male in that population, they're most likely to be fairly close to about that 5'10" mark. Maybe a couple inches taller, maybe a couple inches shorter, but most folks pretty close to that range. So height is one of those beautiful sort of symmetric Gaussian things. Our genes and our epigenetics, or non-genetic but gene expression affecting environment, leads to a really nice symmetric clean Gaussian distribution for that statistic. Not all population attributes, attribute is height, the population is the people. Not all attribute distributions are going to be normal. Sometimes it's an exponential distribution, which means that it's very likely to be the lowest or highest value, and the likelihood of it dropping off, the likelihood of it being any other value drops off exponentially with distance from that most common value. Okay, so that particular distribution looks like a ski slope. It's very common at one side, and it's very unlikely to find anything else. There are Gaussian distributions, there are Poisson distributions, and so forth. There are lots and lots of different, there are multimodal distributions with additional peaks. So bimodal distribution, it's common to have things to the left of the middle value and to the right of the middle value, but not very common to have that middle value. There are multimodal things where it's maybe lots of peaks, and that's normal and okay. That's not noise or error in your data. It's meaningfully multiple different likely values that don't happen to cluster together in the middle. Okay, so when your stats professor is talking about what's the distribution of your data, this is a fairly easy thing for any stats program to compute. It's going to be typically one of the first graphs you produce in a quantitative study. And your interpretation, the decisions you make off of the evident distribution of any particular variable or attribute out of the population, it can tell you to do some things and can tell you to avoid some things. So some of the stats tests that we're trained to use have an assumption of normality. That is, run this test on data sets that are normally distributed. If it's not a normally distributed data set, if it's like multimodal, multiple different valid peaks, instead of one nice symmetric Gaussian peak, then that stats test is assuming you're feeding it normal data, and it will give you a number, but that number is garbage. Okay, so when you are moving past the point of these basic descriptive univariate analyses of each of the variables you collect information on, for those of you doing data collection type studies, the first step is to just kind of get to know your data. What's the shape of my data visibly in terms of its distribution? And those answers will tell you, I can use a multiple linear regression model, or I definitely can't use a multiple linear regression model because these are none of my variables, or none of the important variables, powerful variables, are Gaussian or gamma distributed. Okay, so if you have no idea what I'm talking about, that's okay. The takeaway for this part for univariate statistics and descriptive research is get to know each of your variables before you start trying to charge down the road on advanced sophisticated structural equation modeling and factor analysis and k-means cluster analysis and point clouds and stuff like that. You just want to kind of force yourself to take the time to look at each variable. What is the range? What's the average for this? Is the average the same as the median? Another measure of central tendency that's a lot less vulnerable to strange outliers, like the one person that's 8.5 feet tall that pushes your average closer to the high side. Median is the middle value when they're listed in order. There are tri-means, there are harmonic means, there's modes. What's the most commonly repeated value in that data set? All of these are different ways to get a sense of what's kind of the center of mass of this, what's the middle, but they are different from each other, okay? No data set is perfect in terms of its being symmetric or not too skinny, not too fat, sort of that Goldilocks zone of Gaussian distributions, which would be awesome if it were the case, if we got those, but you know, real data has noise in it. Real data has other factors that we haven't accounted for yet. Some of our statistical analyses will be to tease out what those hidden factors might be. All right, but we can't do any of that more advanced stuff without a real strong grip on the univariate stats. So luckily they're pretty straightforward. And if you're doing a lit review project, you know, you don't have to worry about this, except to get through the tests in 506. All right, so bivariate stats are what most, you know, master's and PhD students starting out, you're probably thinking of. So the other day, you know, last week we were looking at basically a simple bivariate or small polynomial or multivariate linear regression model out of your data. And that's stuff like cross tabulation. What's a table of variable X versus variable Y And a crosstab is just a count of each combination of the values of X with each value of Y. So if we've got 1, 2, 3, 4, 5 as our X variable possible values or levels, we've got A, B, C, D, E as our Y variable values that it could possibly be. How many times do we find 1A and 1B and 1C? How many times do we find 2A, 2B, 2C? And is it sort of evenly spread? Are the counts evenly spread on that table? So this is a two-dimensional distribution. Is it like Gaussian in X and Gaussian in Y, and it looks like a sort of a nice ice cream cone shape? Or is it exponential in X and Gaussian in Y, and it's a sort of weird 3D surface? You really want to know what is the interplay between key variables if your study is asking you to explain or identify what's going on as captured by those variables. So that's a cross tabulation. There's stats tests you can run on that, like a chi-square that says there's no meaningful difference between this cell over here, which looks like it has a pretty different count than the cell up here. They're actually kind of the same within the range of possible combinations. Or it's significantly different. This table is not a flat, even surface when viewed in 3D. It's tilted some way, or it has that sort of weird, sort of strange looking 3D surface when you graph the counts as heights. Correlation is one of the huge things that pretty much everybody's gonna run. They're super quick, they're super easy. Some of the correlation stats tests to say this variable is meaningfully related to and varies in some recognizable way with the behavior of some other variable. As I turn up X, does Y turn up as well? As I turn down X, does Y turn up? So are they directly proportional to each other? I turn up X or increase X and Y increases as well. Are they inversely proportional? When I turn up X, Y decreases. Stats tests are super common. They have been for decades for correlation research. So if you're doing any stats, you're probably doing correlations. And again, you want to know what's the underlying shape of each of the variables that you're putting into a correlation. But you can do multiple correlations where you throw, let's say, 10 different variables together and you see which among those 10 are correlated with each other. One of the functions of stats research is to figure out, okay, so variable 1 and variable 3 are pretty tightly correlated, not because one is sort of influencing potentially or strongly related to the other, but because they're actually measuring the same thing. This is called collinearity or collimation, meaning you think you got 10 variables, each describing some different part of what you're researching, but you've really only got 9. And so you want to pick the worst of the two that is collimated, just throw it out. It's like it doesn't exist in subsequent analyses. Okay. If you put both of those in, it can kind of screw up the model because the real behavior that's getting encoded twice as 2 different variables is hitting a little bit too hard in any subsequent model where they're both being used, if that makes sense. Okay. So part of descriptive research is this kind of stuff. What's the basic relationship between variable 1 and variable 2? Cross tabulation. What is the basic behavior of 2 based on the behavior of 1 or in proportion to the behavior of 1? Correlation models. And then from that, you start to build graphs, right? So once you have a sense of A or variable A and variable B have some interesting interaction, it looks like, let's just throw that on a graph, 1 on each axis, and see what comes out. The main thing that we really want to be careful of, and a good stats professor will help you understand this well, before you get into the nitty-gritty details of specific equations and terms and what statistical thresholds or whatever, all that stuff. They'll help you understand that correlation is not causation. Okay. And we'll have some examples here in a sec to kind of wrestle with that for a sec. Explanatory research is taking that next step with your statistical tests, your statistical tests are going to assume that you understand or that you are demonstrating that variable 1 is driving the behavior of variable 2. They're not just behaving in proportion to each other. They're not just correlated, but there's causality. Okay, so descriptive research, correlation is a description of attributes and how they behave in relation to each other. Explanatory research is saying why. It's because variable 1 drives the behavior, causes the behavior of variable 2, test of causality. And that's explaining why we're getting this dynamic, this phenomenon, this process that we're seeing in nature or in a test tube or out in a field or in people's minds or whatever. Okay, so the key difference between descriptive correlation stats and explanatory causality or causation stats is crucial. All right, a common mistake for students doing any kind of stats-based research is to get an exciting, strong correlation and say, oh, well, X is driving Y. Well, no, they're just related. So for example, it has been observed in verified and repeated scientific research that everyone who eats pickles, everyone who has suffered from cancer has eaten pickles in the United States. They are correlated. There is a 100% chance that you have eaten pickles if you have cancer. Of course, that's silly. Pickles are delicious and non-carcinogenic in reasonable doses, I assume. I'm not that kind of doctor.
Doctor wrong.
Okay, so the correlation is clear. 100% clear relationship between, or likelihood that, if you have cancer, you've eaten pickles before. The mistake would be saying pickles cause cancer, obviously. I mean, this is a trivial, goofy example to make it nice and easy. So the question there is the pickle a necessary cause in cancer? You cannot get cancer unless you eat pickles? Or is the eating of pickles one way to get cancer? Is it a sufficient cause? Okay, so in that case, we would see if pickle consumption did drive cancer rates of any particular cancer, then it could be a sufficient cause, right? Without pickles, and we can test this in a lab or carefully controlled, diet study. Without pickles, we cannot trigger cancer. It's a necessary cause in that case. It is a strong, required, independent variable in your research design in that way. For a sufficient cause, pickles could cause cancer, but they might not. Or pickles could cause cancer. In fact, they probably do, but there are lots of other things that cause cancers as well. Whatever gene or chromosome damage is necessary to trigger cancer and uncontrolled replication. Pickles and drinking motor oil and, laying in a tanning bed too long or whatever, and your DNA get photolyzed by UV light. Those are all sufficient causes, and they might cause cancer each kind of independently of each other. A contributory cause is one where pickles help in your body, or they could help in your body to create the environment for your genetic material to be more vulnerable. So for example, has anybody ever, you don't have to answer this personal medical information, but has anybody ever been on an antibiotic where you couldn't go out in the sun? Okay, that's a contributory cause kind of setup, right? It just makes your skin much more vulnerable to UV damage, and you get these crazy sunburns and sun poisoning, as it's called, when you're on particular antibiotics. And so you just stay inside on those days and you sort of vampire your way through. So we have necessary causes, sufficient causes, and contributory causes And stats tests can help you work out what's the likelihood of each of these cases, but you have to set up the statistical model to test for each of those kind of separately. Okay, so your advisor, your stats professor, they're pretty familiar with this if this is your area of research. They can help you select the specific stats tests you're going to use based on the distribution of each of those variables and based on whether you're trying to accomplish like bivariate or multivariate cross tabulation. You can have two-dimensional tables, you can have three-dimensional tables with extra like slices or layers of the table going upward. That would be a three-dimensional cross tabulation. You can have sort of weird hypergeometry, n-dimensional cross tabulations where you have 5 different variables correlated to each other, and our brains just can't deal with that. So we just look at the numbers instead of trying to graph it out. Or your research is relying on previous descriptive research. You're aware of each of the chemicals in pickles, and you're aware of the photolyzing capacity of sunlight at 1100 watts per square meter at this latitude. And you are throwing those together in various statistical models and different combinations to see which model matches the behavior of the data. And the statistics tell you how well each of those fit. So there are goodness of fit statistics that are used in explanatory research like R squared. R squared is a statistic that tells you what is the percent of the dependent variables, the output, the final observed behavior variables, what percent of that behavior is based on input variable 1 versus input variable 2 versus input variable 3. Very commonly we'll get a situation like, because most things are more complex than a single input variable, we'll have 3 input variables and each is contributory to a problem or a solution that you're trying to wrap your research brain around. One of those 3 is probably pretty strong in its R squared, so something fairly high in environmental research. An R squared between 0, it scales between 0 and 1, an R squared of 0.7, 0.8, 0.9, so 70, 80, 90% of the behavior of the dependent variable rests on this one input variable. But the others, if they are also necessary but smaller, then they might have an R squared of 0.2, 0.3, 0.4. But when you hold that variable constant or you pull it out of the model, you don't get the output behavior. So they're both necessary. One is strong and the behavior of the output varies real strongly with the behavior of that one input, but it also needs the other one present. Okay, so in chemistry, we would call the smaller necessary one like a catalyst. It's not necessary. It's not necessarily what the feedstock material is getting transformed into the output of the reaction, but you got to have that catalyst to make the reaction go. They're both necessary. You got to have the raw ingredient and the platinum or palladium catalyst, or whatever it is, in order to make the new chemical compound. Okay, so your statistical models will say, yeah, you need both of these, or you need both of these in, this is the strength of variable 1, this is the strength of variable 2, and so forth. And add on more variables, add on more nuance to that, and it's multivariate explanatory dose response type studies and advanced statistical modeling. But the fundamental idea is the same. You're trying to figure out the mechanism and use the data to show you which of your mechanism ideas, which of your hypotheses is closest to the correct one. We're never going to get it exactly right, but we can get closer and closer and closer by refining our understanding. Okay, so this is kind of a high concept slide, and so I'm spending a big chunk of class on it because stats are pretty important and can be intimidating. Does anybody have questions about this? And if you haven't taken these stats classes, maybe you're feeling like, I don't even know what I don't know about this, but that's fine. This is a good moment for questions if you have them. Otherwise, we'll move on. Nothing in chat yet. All right, moving on. Cool. If you do have other questions, let me know, of course. So talk through that. skip through that. For causality, in a study where you want to say X causes Y, or you can't have Y without X and Z in some proportion. Maybe it's a sequence of X drives Z and Z drives Y, but without everything in that order, it just doesn't work. Okay, so temporal order, A cannot cause B unless A is before B, which is obvious. But sometimes when you get way down and like programming in equations into your stats software, like, R or Stata or SAS or whatever, or SPSS, the computer will analyze specifically exactly only what you tell it to. Sometimes we humans, we're all guilty of it. We just make a mistake and put the terms in the wrong order, or we run the stats test out of order, and then we come back to it a week later and we haven't had our coffee yet, and we put the cart before the horse in the temporal or the time domain sense. Okay, so A has to come before B. A also has to have some recognizable correlation or association with B. So when A goes up, does B go down, like verifiably? Or when A goes up, does B also go up in terms of its numerical values typically? We talked about necessary versus sufficient. With all real world data, we have to have some kind of a decision rule to take the stress out of the process of what are we going to do with those weird individual values, the 9.5-foot tall person, and there's like one of them ever in human history, maybe, I don't know. And then everybody else is like kind of around 5'10"-ish as an adult male or whatever. Okay, what do you do with those exceptional cases? Do you say, well, you know what? That person is a part of God's beautiful creation and we're going to throw them in the data set too, and it's going to pull our average way off. Or are we going to say, you know what? That person is amazing and exceptional and just not at all characteristic of what we're going after. We're going after the center of mass, what's common and average and most likely. Either one of those answers can be correct based on your particular project and the needs of the research. That's fine. So you want at the outset to have, when you start to get into your analyses, your data cleaning, your error checking and stuff like that, it's that moment where you want to have this conversation with your advisor and ask, what's our decision rule with outliers? Are we going to keep everything in after we've made sure it's not like a typing error, we've caught the one case where we said somebody was 55 feet 10 inches tall. Well, that was just a typo, right? It was actually just 5'10". After we've convinced ourselves and audited our data and cleaned up any transposition typos, stuff like that, are we going to keep exceptional cases in, period? Are we going to throw out all outliers that are more than 3 standard deviations, about 95% of the way out to the end, either side, high or low, of your data set? In qualitative data, you're looking for these exceptional cases. If one person out of the 100 people or 50 people that you interviewed said something that's really different than what everybody else was saying, that's important. You just caught the unicorn. Why did that person say something that's wildly different than what everybody else said? There's something going on there. That's weird. Is it some kind of dynamic in the phenomenon? Is it that person trying to impress you as the researcher, a so-called socially acceptable response bias? That happens. Is it a whole other egg that you just cracked now, and this person is the one person that gave you the window into this whole other side of the phenomenon that you didn't even know about until one person had the courage to speak up about it, or it's normal to them, so they just said, yeah, XYZ. You really want to understand, what's my philosophy, what's my methodology, not my methods, not the recipe, what's my mentality, what's my worldview on exceptional cases for this study? Am I cleaning out everything that's weird? Am I lowering my signal to noise ratio by doing that? Or am I looking for those outliers, those exceptional cases, those weird ones? Because that's where the interesting stuff is, okay? So we gotta have temporal order, A before B, association, A, positively or negatively correlated with B. And finally, having established these first two, third, let's satisfy that we can break the process. We can make it not happen. We can disprove alternatives, other proposed models, okay? And you do this in the stats. You break all sorts of different models and say, well, it's definitely not this. That's a total Charlie Foxtrot, as they say in the military. This model over here, it worked okay, looks about right. This third model, we took this one and we changed it, and it worked, but not nearly as well. Like something's wrong with that model. Let's just, eyes looking in different directions on that one. It doesn't know what page it's on, and so forth. Okay, so we don't really have time to jump into this today, so we'll start wrapping up here. But on Thursday, let's talk about some real world, some studies drawn out of all sorts of different areas of research to try and wrap our heads around this idea of how do we use stats, how do we use qualitative data to, again, we never prove anything with stats. We only break alternatives until there's no other alternative left. Okay, cool. Pack it up for now, and I'll go ahead and post recordings from, I guess, last Tuesday and today on our course website, so anybody who needs to review stuff, you sure can. Sorry that has been lagging behind, but having some trouble with uploads. And keep on keeping on with your outlines, with your annotated bibliographies and your lit reviews. At this point, we're still just generally putting in lots of bullet point level phrases and stuff like that, okay? Cool. Come catch me during office hours or set up another time this week if you need something. See you on Thursday. Thank you very much.
Thank you.
The actual, not the mesh better.